Today we're going to learn about Chapter 2, Section 3, which is about biological macromolecules. Before we begin, make sure that you have your notes organizer. This is going to have a chart on the front with a section for carbohydrates, lipids, proteins, and nucleic acids, and a place for you to take notes over their important information. And then in the center, there's a place for you to summarize information that's common to all four of these types of molecules. So whether you're typing on this or handwriting on it, please take a moment right now to go ahead and make sure you have the notes organizer. Before we begin, let's make sure that we all understand some very important vocabulary terms. These are terms that you have likely learned in the past, so hopefully this is a review for you. But I do want to make sure that we're all on the same page as to what these terms mean. The first one would be atoms. Atoms are what we're talking about with a nucleus of protons and neutrons, and then there are electrons orbiting that nucleus. That would make up one single atom. An element is going to be a pure substance, such as oxygen, carbon, nitrogen. It can't be broken down into any other components. These are the things that are listed on the periodic table. And finally, molecules. The term molecule specifically means you have two or more atoms that are joined together with a chemical bond. And that type of bond is going to be a covalent bond. So molecules are always held together by covalent bonds. To illustrate this, I do have a model here of a glucose molecule. So this is a molecule. Each atom is represented by the sphere, and each bond is represented by this white connector here. So you can see the black atoms, those represent carbon. Those are atoms of carbon that you see forming this ring. The red would be atoms of oxygen, and the white circles on the ends would be atoms of hydrogen. So all of this together is one molecule. It's held together by these chemical covalent bonds, and then we have atoms that make up the molecule, and the different colors represent atoms of different elements. So now let's talk about what we're going to be discussing in this presentation. First of all, our biological molecules, the focus of what we're going to learn about is what are their building blocks, what are their functions, what are examples of where you can find the macromolecule, and then we'll cover also some additional information. You have a place to write all of these things down in your notes organizer. To begin with, let's talk about carbon. Carbon is the basis of almost every single molecule that is important biologically. All of our macromolecule types contain carbon in some form. Carbon is a really important molecule to form the structure of living things and help us carry out the processes that keep us alive. If you see in the diagram here, you'll notice that it has a breakdown of some of the most common elements found in your body, and 18% of your body mass comes from carbon atoms. That is a large portion. It's not as large as the oxygen portion, and that's due to the water molecules. Remember that most of your body is made up of water, which is H2O, so that's why there's so much oxygen. But the carbon would be the second most abundant element in your body. And why is this? Well, carbon has a unique atomic structure. You can see it in the diagram here. Each carbon atom in its outermost energy shell has 4 unpaired electrons. And if you remember from your physical science class, electrons like to occur in pairs. So all 4 of these electrons in the outermost energy level, they want a partner. So that means carbon is able to form a bond where it shares electrons in each of those 4 locations. So carbon tends to form 4 bonds with other atoms. That means it's going to bond very easily with elements like oxygen or hydrogen, nitrogen, and phosphorus. Those are the most common elements we're going to see in these different macromolecules today. Also, because of this versatility in its bonding, you'll notice that carbon can not only form single bonds, which would just be sharing one pair of electrons, but it can form double bonds if it shares two pairs of electrons, or even triple bonds if it's sharing three pairs of electrons. So when we are talking about the chemistry of life, the molecules that are important in living things, there are four categories. And those four categories of molecules are listed here: nucleic acids, carbohydrates, lipids, and proteins. Those are the four macromolecules. Sometimes those are called biomolecules instead. And they perform important functions like important sources of energy, whether you are using that energy immediately or storing that energy for later. They are the raw materials for building different molecules in your body, and they also contain the instructions for making you who you are. So all of these biological molecules, as we mentioned, they're going to be carbon-based, which makes them organic. Organic in the sense of biology means a carbon-based molecule. All organic molecules have to have carbon, and they have to have carbon-hydrogen bonds. So you can see in the diagram, this is a molecule. It's a drawing of a molecule with only carbon and hydrogen. This would be an organic molecule. It has covalent bonds between carbon and hydrogen and carbon, so that makes it organic. Inorganic things would be like salt. NaCl is table salt. This is an ionic bond between the sodium and the chlorine. The chlorine steals an electron from sodium, so sodium has a +1 charge, chlorine has a -1 charge, and that opposite charge is what attracts them together. That is the ionic bond. There's no CH groups, no carbon or hydrogen at all here in the inorganic molecule. Even something like CO2. CO2 is not an organic molecule. It's considered inorganic because it doesn't have any hydrogen in it. It's just carbon bonded to oxygen, so it does not meet the requirement for being an organic molecule. So CO2 is an example of an inorganic molecule. When we're discussing our macromolecules, it's really helpful to break down the word parts. So macro means large. All of these molecules in relation to general molecule sizes are really big, and they are made out of really long chains of repeating subunits. That is known as a polymer. Poly means many, mer means part. So a polymer has many parts. And the parts that make up that polymer are called monomers. So you can see a picture here of a single monomer, one subunit. When you link a bunch of those monomers together, now you get a polymer. So monomers are going to be the building blocks of polymers. All of our biological macromolecules have distinct building blocks that they are made out of. So here's our four categories again: carbohydrates, lipids, proteins, and nucleic acids. For each one, we'll discuss what is the monomer, and then what are some of the characteristics of the polymer that's formed when you bond a bunch of those monomers together. Let's start with carbohydrates. Carbohydrates, first of all, are composed of three elements: carbon, hydrogen, and oxygen. And the thing about carbohydrates, when you look at their molecules and you look at the chemical formula, they tend to have that carbon, hydrogen, and oxygen in a 1:2:1 ratio. A good way to remember this is glucose. If you remember that formula for glucose, C6H12O6, for every one carbon, there's 2 hydrogen and 1 oxygen. Another way of saying that is there's always going to be equal numbers of C and O, and then there's going to be twice as many hydrogens as you had carbon. The building block of a carbohydrate is a monosaccharide. Mono again means one, and saccharin means sweet. So literally one sweet. This is a simple sugar such as glucose, which you see here, a ring-like structure, or fructose, also a ring-like structure, but it's a 5-sided ring as opposed to a 6-sided ring. When we have two monosaccharides joined together, they form what we call disaccharides because di means two. Sucrose, which is just regular table sugar, is an example of a disaccharide, as well as lactose, which is the sugar found in milk. And then if you link together many monosaccharides and make very long polymers, those are called polysaccharides, again, because poly means many. These are going to be your complex sugars, your complex carbohydrates, your starches. Also glycogen, which is stored in your liver. Cellulose, which is found in plant cell walls. Those are polysaccharides, so really long polymers made of a bunch of glucose molecules all linked together. You can see those ring-like glucose molecules that are bonded together. The arrows are showing you the bonds in between the monomers. So we have 1, 2, 3, 4, 5 different monomers that are chemically bonded together to make this one polymer. The function of carbohydrates is going to be primarily energy. This is what your body uses as an immediate source of energy. A simple sugar can give you fast-acting energy. A complex sugar like a starch will give you more long-lasting energy. So if you need energy right now in this moment, you might want to choose a food with more simple sugars like a fruit. An apple, for example, has a lot of that natural fruit sugar in it. That can give you a quick boost of energy. Whereas if you are running a marathon or if you have a sporting event, maybe the night before you will eat a plate of pasta. Pasta has a lot of starch in it. So your body can break down starch over a longer period of time and get a sustained source of energy from that. So energy is the main function of carbohydrates, but there's an additional function of structure and support. The carbohydrates that are in your cell membranes, the carbohydrates that make up an exoskeleton of an insect, for example, can provide support. Also, here's a great picture of cellulose. We talked about cellulose in plant cell walls. That's another structural support function of a carbohydrate. So the food that you eat that would contain carbohydrates would be things like grains, pastas, fruits because of the sugar, vegetables, sugar, honey, potatoes, anything very starchy. Bread would also be another good example of a food rich in carbohydrates. The next macromolecule that we're going to talk about is lipids. Lipids are fats. They're composed of three elements, the same three that we saw in carbohydrates actually, carbon, hydrogen, and oxygen. The difference here is you don't have the same number of oxygen atoms. Lipids are almost exclusively carbon-hydrogen chains. So we don't see a 1 to 2 to 1 ratio. We mostly see a 1 to 2 ratio just of carbon and hydrogen, and there might only be 1 or 2 oxygen atoms in the entire molecule of hundreds of atoms. The thing about lipids is that they are hydrophobic. That literally means water-fearing. That's because they are non-polar. There's no area of negative charge, no area of positive charge, so they are not attracted to polar molecules such as water. Lipids are a little weird in terms of monomers and polymers. They don't really have one true monomer that makes up all of lipids, but they do have subunits that are put together in a predictable way. The basic structure of a lipid is to have a glycerol backbone, which is highlighted in green here, and 3 fatty acid chains. Notice all of the CHs in those fatty acid chains. That's why we're saying lipids are mostly composed of carbon and hydrogen. This type of lipid is called a triglyceride. You may have heard that before. And that's because there's 1, 2, 3 fatty acid chains on a glycerol, hence triglyceride. Lipids can be found in either the solid form or the liquid form, and we classify them based on if they're solid or liquid. A solid lipid is known as a saturated fat. A saturated fat is saturated with hydrogens. It has all single bonds between its carbon and hydrogens. It does not have any double bonds between carbons, so every carbon is saturated with the maximum number of hydrogens. That makes it a solid at room temperature, and that would include things like the fat that's found in meat or butter. Most of those animal fats are saturated fats. These are the unhealthy fats. You don't want to eat too many of them. The liquid fats are the unsaturated fats. There is at least one double bond between carbon atoms in those fatty acid chains. A lot of times there's more than one double bond between different carbons. And these would be the healthier fats, a lot of the plant fats like your oils, olive oil or peanut oil. Those in terms of fat intake are a little bit healthier for you than the saturated fats. The reason why they are liquid is because the double bonds forms these kinks in the fatty acid chains, so the molecules can't pack together as tightly, and therefore they remain liquid at room temperature. So here's a good illustration to show the difference between the saturated fats and the unsaturated fats. Notice in the saturated fat, there's all single bonds here between the carbons. Every last one of them is a single bond. Whereas the unsaturated fat, you have at least one double bond, and that forms this bend or the kink in the molecule. That makes it a liquid because, again, they don't pack together as easily. A primary function for lipids is energy storage. We're talking about long-term storage, not energy that you're going to be using in the near future, like for a carbohydrate. Additional functions, think about the structure of a fat and how it doesn't mix with water. That means it's really good at preventing water loss and creating a barrier. Waxy coatings on the leaves of plants, you can tell that they're waxy if you feel it and you see the water bead up. That helps to prevent water loss. We have our cell membranes are made out of lipids, and that prevents water from flooding into our cells. If we think about the walrus and its insulation, it has blubber, that's a lipid. And so again, here are your examples of the types of foods that would contain lipids. Your butters, your oils, avocados have a lot of healthy fats, nuts and cheese, and then also steroids, steroid hormones or steroid medications, those are lipids. Next, we will talk about proteins. Proteins are going to be the most varied of all of these molecules, meaning there's so many different types of structures, there's so many different types of functions. We could really spend the entire course just talking about proteins, but of course we won't do that. We will try to simplify it into just the must-know information. Proteins are composed of 4 elements. We have our standard carbon, hydrogen, and oxygen, but also notice now we have nitrogen. Every protein is rich in nitrogen in addition to carbon, hydrogen, and oxygen. Proteins make up about 15% of your total body mass. So a lot of the tissue of your body is made up of protein. And that's one of the major reasons why we need protein is because it forms our body tissues. It forms our muscle, our hair, skin, and nails. Collagen, keratin, if you've heard of those before, those are proteins. And proteins, as we will discuss in our cell unit, are made by the ribosome. You may remember that from previous science classes. Now, you'll hear a lot about structure and function throughout the entire course. Proteins probably illustrate that relationship better than any other type of molecule because the structure of a protein is going to directly impact how it can function. The building block of a protein is called an amino acid. There are 20 different amino acids, and you can see them listed to the right side of the screen here. Our bodies can only make about 10 to 12 of those amino acids, so we have to get the rest of them from the food that we eat. Those are called essential amino acids because it is essential that we eat them or else we won't have them in our bodies. When you build a protein out of amino acids, the bonds that hold them together are commonly called peptide bonds. You'll see that word a lot. The word peptide just refers to protein. They're still covalent bonds, but since they are the covalent bonds that bond together amino acids to make a protein, they get their own special name. If you ever hear or see reference to a polypeptide chain, that is simply referring to a protein. So the function of a protein, they do so much in your body. They provide structural support, like the collagen in your skin keeps it from sagging. Proteins help you build muscle. Your muscle fibers are made out of proteins. Signals between cells. Insulin is actually a chemical signal that's a protein in your body that helps regulate blood sugar. They speed up chemical reactions. That is a huge, important function. And those are called enzymes that speed up chemical reactions. We will discuss those in a lot more detail in our next unit. And then proteins are also really important in controlling cell growth. We'll learn about that in the cell cycle unit. The types of foods that are high in protein are your meats or eggs, nuts, beans, fish, cheese, milk. Those are all high in protein. And then in your body, you find protein in the form of hemoglobin and insulin, as we previously mentioned. Our final category of biological macromolecule that we're going to discuss is the nucleic acids. Nucleic acids have 5 common elements. We still have the same carbon, hydrogen, and oxygen that we've seen in every other type of macromolecule so far. We have nitrogen, just like there was nitrogen in proteins. And now we also have a 5th element, which is phosphorus. The 2 types of nucleic acids, you've probably heard of these before, would be DNA, which is double-stranded. You can see that in the image here on the right. That would be a molecule of DNA, and that is found in the nucleus of eukaryotic cells. Then we also have RNA. RNA is the single-stranded molecule that you have over here on the left, and that's found throughout the cell. You may or may not remember the functions of RNA from your previous classes, but we are definitely going to go into more detail about that in later units. The monomer of a nucleic acid is called a nucleotide. Hopefully the similarity in name, nucleic acid and nucleotide, will help you remember to associate those two. A nucleotide has 3 specific parts. There is a sugar, there is a phosphate, which is just phosphorus with oxygen attached, and there is a nitrogen base. Sometimes you may hear that called a nitrogenous base. So in this diagram over here, all along the side, this yellow ribbon part over here, that's where you're going to find the sugar and the phosphate part of the nucleotide, and then the nitrogen bases connect in the center. So this dotted outline here shows you one nucleotide and all of its parts. We have that pentagon, that's the sugar. You see it's connected to a phosphate group, and then it's also connected in the center to this nitrogenous base. The nitrogenous base, you've heard of these before. That would be your cytosine, adenine, guanine, thymine. And again, we'll get into more detail about those a little bit later on in the course. Your examples of nucleic acids, it's really just DNA and RNA. DNA stands for deoxyribonucleic acid, if you remember that term. And RNA would stand for ribonucleic acid. The function of these 2 types of nucleic acid, they really both function in the storage and the transmission of genetic information. This is how information from your nucleus is stored and able to be passed down and used in order to make molecules that make your cells function the way that they do. The last thing that I want to discuss is how these macromolecules are formed. So this is just a brief overview of a general process that forms any type of biological molecule, and that process is called dehydration synthesis. So that sounds really fancy, but all it means is dehydration means a lack of water, right? So we're actually going to remove a water molecule. It tells you right there in the name. And then synthesis means to build. When we're removing the water molecule, the purpose is to build a larger molecule. So look at this diagram over to the right. See how we have this molecule here represented by two subunits with the line holding them together. We have another monomer over on the right that's going to join in. There's an OH on this monomer, there's an H on the polymer that it's being attached to. So by removing those two H's and the O, that means we've removed water, and that leaves behind a bond that needs to stick to something. So it's going to stick to this other molecule over here. And now notice we have a 3-part molecule, whereas in the beginning we had a 2-part molecule and this extra monomer that needed to be attached. Here it is again shown in the diagram at the bottom with a little more detail in molecular structure. So if we have these two sugars and we want to join them together, notice how they both have these OH groups. We can remove two H's and an O. There's our water molecule. And then the O that's remaining, this guy right here, needs to bond to something. So what does it bond to? Well, it bonds to this other sugar. So now we have two sugars, a disaccharide, whereas we started with two monosaccharides. So again, we're building a molecule by removing a molecule of water. This requires the input of energy. Whenever you build molecules in biology, you have to put in energy. Think of it as if you were building a shed in your backyard. If you have a pile of wood laying on the ground, you have to put energy into it in order to build a larger, more organized structure. And so when we use our macromolecules, when we digest them, for instance, we have to have a way to break them down. This will be the opposite of dehydration synthesis, and it's called hydrolysis. Hydro means water, lysis means to split. So now we're adding water, and the water is going to attack this bond right here with the help of an enzyme. And what the enzyme will do is it takes the water and it puts one H over here, it puts the OH over here, and now we have two molecules where we only had one to begin with. So you can see it's the exact opposite of what we just saw in dehydration synthesis. That image down at the bottom shows you that disaccharide, what happens if you add water, how the water attacks that bond and forms it into two monosaccharides. So that is the end of our presentation, but I want to revisit that note sheet. This is the back of the note sheet. So on the front, if you printed it double-sided, you have this graphic organizer. And then on the back, there are some practice questions. It's really important for you to reinforce this information that we just discussed by answering some practice questions here. So I would recommend reading each of these statements, and for each statement, see if you can answer it or fill in the blank based on memory. If you get stuck, there is a word bank over here that you can use as your choices, but really use this as a study tool to see what you remember first before relying on that word bank. So once you go through these practice questions, if the answers to all of these questions make sense to you, then you really have a good understanding of what we discussed today. If they don't make sense to you, please make sure you ask for help either in class or during a help session so that you can understand this information. So I hope this was helpful, and if you have any questions, please feel free to reach out.